Hi there, and welcome to this video on A-level biology for the AQA specification, focusing on the topic of cells, and in particular, transport across cell membranes. I'm Manisha from StudyMind, where we help you to revise A-level biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, please make sure to click that subscribe button. And whilst you're watching, feel free to leave any comments down below of anything you're unsure about, and let us know if it's your first time watching so we can send you our free revision resources. We also have helpful timestamps to guide you through the specification. So, let's get started.
Welcome to lesson 9 of 9 in this tutorial, covering transport across cell membranes. This is the final video in our series of 9 lessons on the topic of cell structure. Here are the key specification points for this lesson. The first is to look at simple and facilitated diffusion, osmosis, and active transport. Next, we will cover absorption and co-transport. In the last lesson, we looked at cell membranes and the structure in prokaryotes and eukaryotes. Here are the AQA specification points for this lesson. Feel free to pause the video now and read through them before we begin. We'll start by looking at movement via simple diffusion. Net movement of particles down a concentration gradient from a high to a low concentration is known by diffusion. A concentration gradient is the difference in concentrations of a substance between two regions. These two regions are often divided by a certain distance. Diffusion occurs until dynamic equilibrium. A particular substance will diffuse from a region of higher concentration to a region of lower concentration until the concentrations of both the substances in both the regions is equivalent. This is known as dynamic equilibrium. Diffusion is a passive substance. It does not require energy. Particles continuously move around randomly, and they spread out until equilibrium is reached. Learn the definition of diffusion word for word. It is important to use the word particles and the word net, for the reasons shown here. Now let's fill in a table affecting the factors for simple diffusion. The first thing we'll be looking at is the size of the concentration gradient. We know that the higher the difference in concentration, the higher the diffusion. Next, we'll look at the diffusion distance. We know that the rate of diffusion is going to be inversely proportional to the distance. Our next factor is going to be surface area. The higher the surface area, the higher the diffusion rate. Our next factor is molecular size. We know that smaller molecules will diffuse more quickly. Next, we'll be looking at temperature. We know that the higher the temperature, the more kinetic energy the particles have. meaning that the diffusion rate is going to be quicker. Finally, we will consider stirring. This applies to experiments, but will increase the diffusion. The membrane has to be very thin to provide the shortest path length for the substances to diffuse across. Small, non-polar molecules can simply diffuse through a phospholipid bilayer. Ions are polar molecules. Lipid-based molecules are non-polar. Very small molecules and ions are usually small enough to just diffuse through the phospholipid bilayer. Now let's look at facilitated diffusion. This is the transport of polar molecules and ions into and out of a cell, involving the use of carrier and channel proteins. Polar molecules cannot pass by simple diffusion. They cannot cross the hydrophobic barrier, so instead will travel via facilitated diffusion, and polar and hydrophilic molecules will travel through carrier and channel proteins. They are facilitated across. The channel and carrier transport proteins are structurally designed to interact with polar molecules. Transport proteins have pores that are filled with water to attract hydrophilic molecules. The proteins are polar in this pore region. They can readily interact with polar molecules and allow them to travel through these pores to get into or out of a cell. Next, we'll look at channel proteins, which are less selective, and they mildly discriminate between molecules based on size and charge. By contrast, carrier proteins are more selective and only allow one specific type of molecule to cross. Now let's fill in this table to recap what we've just learnt. The first thing we'll look at is simple diffusion factors. These will also apply to facilitated diffusion. Next, we will look at the number and the size of transporters. The more transporters we have, the faster the rate of diffusion. Next, we will look at regulation by the cell. The higher the temperature, the more kinetic energy the particles will have, meaning the faster the rate of diffusion. Our next specification point will cover the movement across membranes by osmosis. Osmosis is just about water. Water is a polar molecule, so it diffuses across membranes via facilitated diffusion. Aquaporins are the water transporters. They are special channel proteins that facilitate the diffusion of water. In equilibrium, as there is more salt on the right-hand side, the volume of water will be greater. Here, we can see that it's the concentration of water that increases, not the volume. Osmosis is driven by the concentration gradients of water as well as water potential. The greater the concentration of water in a membrane, the more kinetic energy the system has. Hence, the greater the water potential. the pressure exerted by water molecules on a membrane is measured in kilopascals. We use the Greek letter psi as a symbol for water potential of a solution. The solute potential is a measure of the extent to which the solute concentrations lower the water potential. Pressure potential is a measure of the pressure that develops inside of a cell as a consequence of the inflow of water. We can use these three in the equation shown here. Water potential is equal to pressure potential plus solute potential. You don't have to worry about the solute and pressure potential too much. Just learn their definitions so that you can use them to define water potential in general. The water potential of pure water at atmospheric pressure is going to be 0. Adding solutes, such as salts or glucose, reduces the concentration of the water per unit of solution. The more solute added to the water, the more negative the water potential of the pure water will become. In osmosis, water moves down a water potential gradient across semi-permeable membranes. Water will move from a region of high water concentration to regions of low water concentration. Adding solute, such as salts or glucose, reduces the concentration of the water per unit of solution. Hypertonic and hypotonic solutions have different water potentials. However, both sides are said to be isotonic. Now, we will cover the movement by active transport. Normally, molecules diffuse down a gradient. In most cases of transport, molecules spontaneously move along their concentration gradient. That is, they naturally move from a region of higher concentration to one of lower concentration. This is an active process using ATP. In many cases, cells have to transport molecules from regions of lower concentrations to higher concentrations. This moves against the concentration gradient, needing ATP. Active transport allows cells to store substances that would otherwise easily diffuse out of the cell. This can be used to remove waste products. Active transport requires the hydrolysis of ATP. It is active as moving particles against a concentration gradient always requires energy. Therefore, active transport relies on the hydrolysis of ATP in order to produce ADP and an inorganic phosphate. Diffusion, osmosis, and facilitated diffusion are all known as passive processes, as they do not require ATP to occur. Active transport, in contrast to these, is active. Active transport is carried out by carrier proteins or protein pumps. Every carrier protein is unique to each type of molecule that needs to be transported across a membrane. The sodium-potassium pump is one of the most common carrier proteins. Let's fill in this table about the components of cell transport. The first type we'll be looking at, in active transport, we use carrier proteins. In facilitated diffusion, we also use carrier proteins. In simple diffusion, we simply have diffusion through the bilayer. And osmosis requires the use of aquaporins. Next, we know that the process of active transport is active. Facilitated diffusion is passive, as are simple diffusion and osmosis. Next, let's look at the concentrations. Active transport moves from a low to a high concentration. Facilitated diffusion will move from a high to a low concentration, as will simple diffusion and osmosis. Diffusion is mainly for small molecules. Cytosis is for large molecules. Larger molecules usually have some difficulty crossing a membrane. Exocytosis is when a vesicle containing molecules of substances fuses with the inside of the cell membrane and the molecules are secreted from the cell. Endocytosis is when the cell membrane binds to a large molecule, resulting in the formation of a vesicle. The vesicle contains the large molecules, and this will enter the cell. Phagocytosis is endocytosis, which allows for the uptake of solids, whilst pinocytosis is endocytosis, which allows for the uptake of fluids. Our next point is to look at co-transport. The small intestine is a major site of nutrient absorption for digested food. The ileum walls are lined with millions of villi. Villi are small structures whose inner surfaces are folded. Through being folded, the villi increase the surface area and rate of nutrient absorption, for example, of glucose and sodium ions. The villi are lined with folded epithelial cells. The upper layer of these epithelial cells, which is the surface that comes into contact with the digested material, is folded to increase the surface area. One surface of the epithelial cells has microvilli to increase the surface area further. This can be seen here. Absorption of the products of digestion begins in the duodenum. Majority of the absorption will occur in the ileum. Specifically, absorption occurs at the microvilli surface of the epithelial cells which make up the villi. Let's go through the route of nutrients in absorption. First, the nutrients are absorbed from the lumen of the ileum through the villi. Then, they travel into the epithelial cells of the small intestine. The nutrients will finally reach the bloodstream. From here, they go into the hepatic portal vein or into the lymphatic system. This diagram visually displays the steps we went through before. The nutrients are absorbed through the villi into the epithelial cell and then they go into the bloodstream. Glucose travels from the lumen of the ileum to the bloodstream through the sodium-glucose co-transport mechanism. This uses co-transporter proteins. Let's look at the steps of sodium-glucose co-transport in some more detail. The sodium-potassium pump will actively transport sodium out of the epithelial cell and into the bloodstream. This allows potassium to come in the other way. This means that the sodium concentration in the epithelial cell becomes lower than the sodium concentration in the ileum. Therefore, sodium will move from the ileum into the epithelial cell, down the concentration gradient via facilitated diffusion. It will be co-transported along with glucose. Then, sodium and glucose will diffuse through the epithelial cell and out of the basolateral membrane into the blood. Glucose will travel through a GLUT2 transporter. water is absorbed via aquaporins and relies on the sodium movement to create an osmotic gradient. Here's a step-by-step summary of the absorption of glucose and sodium. It's quite a complex process, so feel free to re-watch this section again slowly. Our final specification point is to look at the adaptations of cells for rapid transport. First, the villi will increase the surface area in the ileum and hence increase the rate of glucose and sodium transport. We saw earlier that the higher the number of channel proteins, the faster the facilitated diffusion will be. Similarly, the more carrier proteins there are, the faster the facilitated diffusion or active transport. As the number of aquaporins increases, the faster the rate of osmosis becomes. Here's a summary of all 4 factors affecting the rate of transport. We've now covered all the learning objectives for today's lesson. Feel free to pause the video now and re-watch any of the sections you feel unsure about. Let's try a little pop quiz to test our knowledge of this section. How is the phospholipid bilayer arranged in the cell membrane? In the bilayer, the hydrophilic heads will face outwards and the hydrophobic tails will face inwards. Which of these do carbohydrates not play a role in? Transport of ions across cell membranes is one of the only things that carbohydrates cannot do. Finally, which of these does not affect the rate of simple diffusion? ATP is not required for simple diffusion, therefore it cannot affect its rate. Well done! We've come to the end of this topic on cell structure. We've now completed Lesson 9.
If you enjoyed this tutorial, make sure to subscribe by clicking down below and leaving a comment of a topic that you'd like to see a video on. Click here to watch the rest of our videos in our A-Level Biology series, or visit our website studymind.co.uk for past paper compilations by topic and specification.